Okay, thanks a lot, Florian. It's really a big pleasure for me to be part, and thank you for organizing all these things so uncomplicated and without any further delay. Let me just jump into it. We are very happy that we also contribute here in our joint research activities with the SFB here in Vienna, in particular also with Florian research activity. And from that perspective, I decided to talk really about the world of reprogramming cells in the central nervous system, with a particular focus on human stem cell biology. And that is somehow very important because, as you all know, the general source of um, pluripotent cells that we use in stem cell biology for modeling biological function and to gain data from models in cell culture comes from embryonic stem cells. And these are very, very beneficial in order to get all the different cell types which you need for the derivation of all existing cell types in the human body. I just try to get my laser pointer here. Let me check. Yeah, it's obviously not that easy to get a laser pointer, and
Sometimes we can also see the mouse, but now we don't see anything.
yeah, do you see my mouse now?
I don't think so.
The cursor? Yeah, what is this here? Nächste Folie, Timer, Live-Video. So this is a new version of Zoom. I cannot. Maybe I tried without, and will let you know where I focus to. That's anyway the overview, yeah, which I call really the periodic table of cell and cell biology. You see all the cell types that we can derive, but the real important and I think a revolution during the last 2 decades was that one can use reprogrammed cells as well, not starting from embryonic stem cells, but getting mature somatic cells into a state where they are very similar or maybe even identical to embryonic stem cells. And that is something that you, of course, know from textbooks because it has been started many, many decades ago, primarily by the research of John Gurdon's lab, who did this with frog cells and were able, as you can see here in panel B, to reprogram somatic cell nuclei just by transplanting them into enucleated oocytes and getting them reprogrammed to an embryonic state that make it possible to get cloned animals out of this technology. That was, the first application of de-differentiation, as it was called at that time, and reprogramming in an animal context. And as you know, that has been then realized about 20 years later in mammalian, first in sheep, where clone sheep dolly was generated by the same technology. And we have in the meantime know that more or less all mammalian animals can be reprogrammed or regenerated or cloned by this somatic cell nuclear transfer technology, which was founded then at the end by John Gurdon and people from the Rosslyn Institute in Scotland. What this told us was at that time that the oocyte contains factors, bioactive molecules that are able to drive this cellular transition to make out of somatic states that are imprinted in the genomic loci in naive state, which allows them to develop into pluripotent embryonic-like stem cells, which have been then named correspondingly reprogramming factors, although one did not know what these factors are. And nowadays, we know that this cocktail, how it could be composed, it's no more an unidentified heterogeneous mix from the ooplasm from enucleated oocytes, but we have an insight into a molecular composition of something like a reprogramming cocktail. Although one should be cautious, because that what I will now tell you and what we know as Yamanaka factors is representing one possible cocktail of reprogramming into pluripotency, but there might be different from that what is occurring in the ooplasm after somatic cell nuclear transfer. But the outcome is the same. Yeah, we see reprogrammed cells, and we know this quite better. And therefore, let us briefly look at that. Although I guess most of you know what the Yamanaka factors are and how they were identified, but let us give us a few minutes in order to really recapitulate the history and the biology behind from the basis of transcription factor networks. So one of these Yamanaka factors belongs to the POU transcription factor family, so the abbreviated POU, which is an acronym and stands for the origin of the first a description in the literature of the transcription factors, and one stands for the octamer domain. So it's a transcription factor which belongs to the homeobox family and has a very interesting expression pattern, which you can see here from an historic publication from the 90s of the last decade, where people have shown that it's been enriched in the inner cell mass during the embryonic development. That, for example, when you see in this in situ hybridization, the upper panel in the bright field, we see that we have a very strong black signal in the inner cell mass, but not that much in the troph ectodermal cells, so this outer layer at the blastocyst, and we still have it in the hatched embryos, in particular then at day 5.5 mouse in the X cylinder, so where we have this still pluripotent epiblast-like cells that can give rise to all different tissues in normal physiological development in mice. Interestingly, after implantation and gastrulation, the expression, or here the abundance of the mRNA, drops dramatically and goes to zero in all tissues, with the exception of primordial germ cells, as you see here at day 10.5. There are then very rare cells in the genital ridge which reactivate the OCT4, or they keep it. And this will be then on until the time point of fertilization, where again male and female cells will come together, and where we then will have already maternal transfer of the protein and the mRNA, which then again, and here closes the circle, will be then result in high abundance of the transcript, but also in the protein in the early stages of embryonic development. So obviously this factor is correlating, or the abundance of the mRNA and the protein is correlating with pluripotency. And that is here also depicted on the left-hand side by antibody staining. Yes, we see in undifferentiated cells this POU factor OCT4 highly expressed, but not in differentiated ES cells. And again, the in vivo side on the right-hand side, we have an OCT4 signal based now here on protein only in the inner cell mass, and highly enriched, and no more than after implantation. What is really interesting, that OCT4 is not only part of the Yamanaka cocktail, but the genetics and phenotyping behind it, because people, when they identified that OCT4 obviously plays a role in the natural establishment of stemness identity in mouse, and they reasoned that it might be, of course, also true for human cells. Of course, they genetically manipulated OCT4 in mouse, yeah, by generating knockout mice, yeah. And for that, the normal procedure is that you do this in embryonic stem cells in heterozygous versions, and then you get out of these gene targetings, at least with classical conventional gene targeting methods, the normal heterozygous animals, which you then intercross, and from the intercrosses you would then expect to get by Mendelian ratios the homozygously deleted, the wild types, and the heterozygously deleted. But this expected and the expected outcome was that you might not get homozygously deleted, yeah, because it's highly expressed. But that was not the case, interestingly. Yeah? You see on the panel top left-hand side an in situ hybridization against OCT4, and we see compacted morula. These are these very tightly packed cellular structures, which are partly containing a signal. So obviously we have an OCT4 message, and these might be wild types or heterozygotes, but we see definitely also early embryos at this compacted morula stage, which is about 2 days after fertilization, which appear to be absent of an OCT4 signal, which is somehow strange. Yeah, you can delete it and obviously say they can still develop into morula. And it's even more apparent if you now look on the right-hand side at the bottom, if you look at 1.5 days later, where the blastocysts have been formed, that can be all done ex vivo in vitro. Out of the morula, you have the formation of the blastocysts with the trophectodermal layer, which is necessary for the implantation step, and the inner cell, and here it's now stained with an antibody. And here again, we have blastocysts which look normal and have an OCT4 signal only in the inner cells. On the left-hand side, these 3 blastocysts, but there's obviously also nicely looking blastocysts which are devoid of OCT4 signals. So one can generate, at least until pre-implantation, OCT4 deficient embryos, and they do not show any phenotype. Yeah, they have not any sort of defects, which is remarkable because the OCT4 is highly expressed at earlier stages, but obviously it does not have a role, an apparent role at least, when it comes to the analysis of the pre-implantation embryos. The real striking phenotype is when you try to take these genetic variants of OCT4 and try to get mouse embryonic stem cells in culture. For that, you would remove the trophectodermal cells from wild type. You see on the left-hand side the example, and we see colonies growing out of that, and that will be the birth of a new embryonic stem cell line out of the inner cell mass of a blastocyst. And if you do this from the normal-looking homozygously deleted OCT4 embryos, you see not outgrowths of these inner cell mass like embryonic stem cells, but we see this very characteristic trophectodermal giant cells, which are reflecting a trophectodermal lineage, but no more pluripotent stem cells, meaning the phenotype of the OCT4 deletion results in formation of trophectodermal cells and no stabilization of pluripotent stem cells. And that means that OCT4 is necessary for the establishment of pluripotency, which is obviously a very transient aspect in vivo, but being stabilized can be stabilized ex vivo in vitro by cell culture, and no more able if you do this by with the OCT4 deficient embryos. So OCT4 is a major player in the establishment of pluripotency stem cell functions. And the same would be true for, and I don't want to go too more deeper into that, but for other Yamanaka factors or potential reprogramming factors, and in particular, the SOX2 protein transcription factor is important here, but also others, and they bind usually as heterodimers to DNA to corresponding consensus sequences. You can see here 2 types of those. On the left-hand side, the FGF4 type, on the right-hand side, the UTF1 type binding site for the heterodimer of OCT4 and SOX2. The OCT4 is given binding or component is given in green, and the DNI binding component of SOX2 is showing in blue. And on the right-hand side, blue, the HMG belongs to the SOX2, the blue one, and the purple one on the right-hand side is OCT4. So, and one has identified by genome-wide analysis how these transcription factors play together in order to control a stemness program. ChIP-seq analysis, so chromatin immune precipitation analysis, where you systematically screen for bound genes by the transcription factors are identified around 300, 3 to 400 loci where these bona fide stem cell factors bind to in a joint manner. And out of that model has been proposed that these three transcription factors, so the OCT4, SOX2, and NANOG, build a hierarchy of three factors that have control over these hundreds of stem cell genes, which are necessary to be either activated in order to get this unlimited proliferation, and on the other hand, block differentiation genes to keep cells in a naive, undifferentiated cell state. Yeah, and that was the basis for Yamanaka's finding now, 20 years ago exactly. It has been published, and there will be an anniversary this year in fall in Kyoto, where Shinya developed with a student in the lab the systematic identification of the Yamanaka factors. And actually he did this just because it's the 20th years of the identification. I will briefly talk a few minutes about that, how are they identified, because it's so an elegant experimental setup. So the starting was that they decided to just, with an educated guess, focus on 24 factors. Our three major transcription factors included into this list, but also other factors filling up this 24 factors. And their hypothesis was, when we overexpress these genes, which are no more expressed in somatic cells, they will acquire embryonic-like phenotype. And for that, getting this 24 genes in parallel being overexpressed in mature differentiated cells, they used a retroviral system where they packaged the cDNA of all the 24 factors into a transgenic retroviruses. And then they placed this in an experimental setup where beta-GU gene was under control of the FBX15 locus, which is down in somatic cells, no transcription at all, but very high transcription levels in pluripotent cells. And then they reasoned, when the very rare reprogramming event upon the activation of the 24 factors will occur, we can select this out by antibiotics because the beta-GU will be activated, which allows the selection of G418 resistant colonies cells because it's been reactivated by the Yamanaka factors, and then the pluripotent cells will be formed. And indeed, somehow crazy experiment, yeah, overexpression of 24 factors by retroviruses in parallel in one cell line, and they used mouse embryonic fibroblasts for that, and indeed could show just by morphology, first of all, that these cells really develop features as you know them from inner cell mass like embryonic stem cells. So you see the MEF cells, so the mouse embryonic fibroblasts on top right-hand side. These are the starting populations, these characteristic sickle structured cell building a monolayer, and the colony forming very small cells, embryonic stem cells on the left-hand side. And in the middle, you see what happens if you overexpress with 24 factors in parallel in the MEF cells. They also form these kind of characteristic colony-like structures, which are reminiscent to that of the embryonic stem cells. Also, the proliferation goes up. They do not show more senescence as normal MEFs show, as you can see on the bottom, because the cell numbers rise like ES cells. And also the transcription program, which is here being investigated just by qPCR of some candidate genes, show that they acquire a little bit more profile of embryonic stem cells. And therefore, they have designated these cells like induced pluripotent cells or iPS cells. And yeah, to bring this story now to an end to the Yamanaka factors. They very elegantly used an n-1 approach to break down the real essential and necessary factors out of this 24 to the only 4 factors which are needed. And they did this just by omitting individually only 1 factor out of the reprogramming and then checking the colony numbers which are being formed. And as you can see in this panel A, if you omit factor 2, there's almost no change in colony numbers. But if you omit 14 or 15, there's no more colonies. So obviously 14 and 15, in contrast to 2, has a major effect on the formation. Yeah, and they concentrated now on the 10 most strongest factors and did another reprogramming when with the 10 factors only. And here again, n-1, 14, 15, strongest effect. That results then to a 4 only factor protocol. And here turns out that the CMYC KLF4 SOX2 on OCT4 is sufficient and necessary with the exception of CMYC. One can leave this out. But 3 plus CMYC is necessary and sufficient to induce full pluripotency in treated somatic cells, which can be very nicely and robustly finally experimentally validated because Yamanaka and other colleagues then later could show that from these genetically manipulated cells which have never undergone sexual reproduction, nothing like normal way of embryo formation, with these cells, you can get offspring, which is, of course, a bona fide proof of pluripotency, at least for mouse stem cell biology. And that for sure deserves then the award of Shinmei Yamanaka and John Gurdon by the Nobel Award 6 years later than 2012. Yeah, what this is now the status that one can reprogram cells. By this relatively simple genetic manipulation over expression of just 4 factors by viruses. Meantime, new methods gained importance. I don't want to go into that. That's commercially available by kids, but also by more classical transgenic manipulations, but they all have in essence in common that you overexpress 3 or 4 factors in any sort of somatic cells, and then you get these fabulous iPSCs. And here is shown how we usually do this when we start from a skin biopsy, where here a control individual gives a skin sample by a punch biopsy, which is 3 millimeters inside. This little sample will then come into cell culture, where we then generate fibroblasts out. And these fibroblasts grow in culture. We transfuse them with the 4 factors. And then after 2 to 3 weeks, we see formation of these colonies, which are here marked by fluorescence colors, here in green for mature pluripotency. And then we isolate them and can get them into culture. So it's really a relatively straightforward and simple experiment to get these iPS cells out of the patient. And now with that, we can do all the fine, nice things as I showed you at the beginning, get all specialized differentiated cells that might be of high interest for you or desire to be investigated. So, for example, think about cell types that you want to analyze in order to understand a disease which are hardly accessible, like an Alzheimer neuron, yeah? Or think about cardiomyocytes that are very hard to be biopsied from patient, but are have a dysfunction which is coming from a variant, from a mutation in the patient. You would like to understand the disease in cell culture, but you cannot get really access to primary tissue of the patient, usually only post-mortem, but then, of course, it's very hard to analyze early stages of the disease. And now with the iPSCs, you have a completely new way of access to it. A new way of access to it. You can bring these cells in an unlimited human version into cell culture and now can analyze potential dysfunction. Yeah, in this case, we see in a an electrophysiological aberrant response of cardiomyocyte cells in culture that can be measured by patch clamp techniques. Obviously, just morphometric differences of neurons here in the neurites, as you see synapse formation in terms of neuronal cells. So one can really analyze those phenotypes ideally with the respective cell types with a human model, which carries the corresponding mutation. And by gene editing approaches, one could make a wild type like control where we repair this mutation and we see the direct difference of the phenotypic effects from wild type where they see the variant. And if you have a phenotype that you can measure, one can then also screen, as you can see in panel B, with candidate drugs or even libraries of small molecules, whether or not you can revert the phenotype. And that would then bring the whole drug validation screening system into the cell culture dish, where the human preclinical model is already integrated into the search for key targets and the corresponding small molecules that target the targets directly in cell culture in the human model from the beginning on, which is usually not the case, where you need to work with preclinical models, usually with HEK cells, so cultured cells which are far away from the cell type, or even with mouse models or any other animal models which also do not fully recapitulate the features of the human physiology and biochemistry. And at the end, what also is given in C, it has a major importance for checking undesired side effects of drugs when it comes to toxicity particularly in the cardiac and nervous system and hepatocyte system, because many drugs, for example, new psychiatric drugs, which have a positive effect in the nervous system, might have negative unwanted side effects in the cardiac system. And that is something that one could pre-test preclinically in the cell culture dish by this technologies without going into human phase clinical 3 and 4, where the development of such undesired side effects would be extremely costly because the drug is already close to the market. Yeah, what I, of course, want to share with you is not only the classical reprogramming in the neural system and the potential application, but also the new developments, how reprogramming can be now extended or brought to second and third generation reprogramming in order to get even more flexibility for developmental studies or for translational approaches, which is somehow entitled by direct reprogramming or lineage conversion. And in order to explain this to you, it's good to talk about the mechanism of cellular reprogramming. So to understand what's happening during the conversion and the change of cell type from fibroblast cells to an pluripotent stem cell type. Yeah, and the mechanism is being subject of experimental analysis now from the beginning on of IPCs. It's still not completely uncovered, but I share with you the current status or the models that has been discussed and are also partly experimentally validated by researchers. And it started already with Yamanaka's interpretation to explain the fact that, first of all, the reprogramming needs quite a lot of time, 2 to 3 weeks, particularly with human cells, and also the efficiency extremely low. Yeah? If you would calculate the number of input cells with the colonies reflecting the number of generated iPS cells, it's in the range of 1 in 10,000 or 1 in 100,000. So really very, very rare cells. And he explained it by the fact that there might be elite cells present in the cell population, either from beginning on, that would be the predetermined model, or it would be stochastically induced by transcriptional noise in the system. And that could be, for example, if you reprogram from skin, you must anticipate that your population of cells is not 100% pure. There might be some contamination of, in this case, the skin-derived fibroblast with skin stem cells, very low contamination participation or contribution, but these yellow cells might be the real cells which are the target of the reprogramming. And these elite privileged cells will make it to the red iPSCs, and there might be some of those where the reprogram process might be not fully executed, and therefore we have this only half red cells. Yeah, and that might be the explanation why only the rare cells become iPSCs. And this is also true for the induced elite model, where by transcriptional noise, although all the cells are identical, similar, there's always a little bit of deviations in terms of transcriptional noise occurring that makes only a subset of numbers of cells eligible for the real reprogramming. The other model, on the other hand, would propose the stochastic model that we induce by the overexpression of all four Yamanaka factors as a stochastic change in the population, and the stochasticity or the random changes of cells just comes from the fact that you do not have a full control over the activity of all four factors. The stoichiometry, the length, and the overexpression levels is a subject of random activation, and only in very rare cases you have the right setup in terms of time and expression level that cells make it to the red stage, and that would be the stochastic model. And of course, if you think in terms of gene regulatory networks that takes place during this transition from blue via yellow to red, it would be very interesting to look at. Yeah, we really would like to understand what is happening here when we induce OSKM. So these are the four Yamanaka factors in fibroblasts, and we check at a single cell level what happens in terms of changes of morphology of cells, but also changes in the transcription networks that the OSKM is activating in a reprogramming setup. And on a morphological view, it's very unclear what happens, because during the first early days, almost a week, most of the cells stay with their fibroblast phenotype at a morphological level. Some of them undergo apoptosis, senescence, or transformation, but there's definitely no way to see some sort of pre-pluripotency or pre-stemness during that phase. In order to get insight to that, one, of course, needs to get insight at the transcriptomic level, at the single cell level. And that is somehow the analogy to an philharmonic orchestra. Yeah, when you hear this wonderful sound of music that needs to make it in the composition of all individual instruments, and only when everything comes at the right time point with the right voice together, conducted by the conductor, then you see this sound in harmony, which shows that everything was right. But sometimes you hear a noise, sometimes the sound is not good, and then you need to dissect this in more detail. And that would start somehow in this analogy that the conductor will ask that only the violins should play their voice, and maybe only the violas, the celli, and so on. And maybe he will identify that there's something wrong with the violas, and then maybe even individual player needs to play, and then the conductor will see that's only one who makes here the difference. And that is actually something that one has to look at also during reprogramming in a way. Yeah? We have a very, very complex interplay, which has to come into harmony when they do all the things at the right time point. And in order to understand this, we cannot work with the classical transcriptomic analysis, because we would lose the information of this individual, in this case, harmonic interplay or the transcription network activity that needs to be activated in order to become productive iPS cells. So it's imperative nowadays that one can analyze this by single cell analysis, where you capture single-cell transcriptomes from all the cells in this heterogeneous population of cells, and then analyze this individual voices or instrument groups, here now schematically shown by just 4, and then you capture their transcriptomes. And then, for example, you might see that some of these cells, so particularly the yellow, purple, and blue ones, they are more or less reflecting the bulk transcriptome, so they have more or less a similar or identical transcriptomic profile, but there are these green ones which are completely different. Yeah, they have a different contribution to the overall success of the music, or in this case, the reprogramming. And then one would look at this green cells in more detail, what is happening here, and then trying to understand which kind of gene activation network programs are going on in this population of cells. And that is more or less what people have been carried out. As said, it's still an open story, but there's several in-depth analysis being published to shed some light on the reprogramming chronology of events analyzed by single-cell technology. And for example, what has been found out is that at the very early beginning, we have a stochastic phase. That is an initiation phase where, first of all, proliferation is being increased. We have metabolic changes. We have initiation of MET, which stands for mesenchymal epithelial transition, something which also occurs during some developmental processes. We have changes in epigenetic marks. And but that comes in a very stochastic manner. It's not really being known which kind of change will be very beneficial for at the end to form iPSCs then at the end. Single cell analysis showed that we have this at the beginning, stochastically randomized, but the very last stages undergo very deterministic or hierarchical phase, which is being started by the activation of endogenous SOX2 gene. So the endogenous gene in the reprogrammed cells are being reactivated at a SOX2 gene at a relatively late stage, and consequently this has then an effect on target genes, here, for example, by Lin28 or ESRB being activated, which in turn then activate other downstream genes where OCT4 is part of. And that comes not stochastically, not in a randomized manner, but really follows a chronology of events which would have anticipated from reporter and ChIP-seq analysis, where one exactly see this hierarchy of transcription networks. And if then SOX2 as an starting event is being initiated, we have a very high probability that at the end of these transitions, there will be a productive formation of iPSCs. Yeah, and in the intermediate phase, although there is some indication that still the metabolic effects are important, play a big role here. It's not really being known how you can define to increase, for example, or to get precursors of the SOX2 positive cell population. It's still an unknown fact. There must be a rate limiting step which out of the stochastic phase defines those kind of cells here shown in green, who might be necessary to get to yield then in the SOX2 positive cells, which are necessary for the formation of iPSCs. There are some candidates being discussed, but it's still an unsolved question what happens here. But what one learns from here is very important. Yeah, we have the stochastic phase, which is more or less a destabilization phase, and it comes with changes in histone marks, activation of DNA repair. So these kind of things which keep the genome stable in an epigenetic and genetic manner, they are weakened. So we have a destabilization in the first phase. Whereas in the last phase, the deterministic phase, we have more or less a classical developmental program where transcription factors play their program, and that results in, at the end, in a complete reset of the phenotype, which, for example, can be reflected by cytoskeletal remodeling, because these cells, the iPS cells, usually by morphology look completely different from the starting cell population as fibroblast cells. And we reasoned some time ago whether we can separate these two functions from each other. You know, this destabilizing phase from the lineage specific hierarchical phase. And we thought that OCT4 might be necessary, or might play a very important role in that, because OCT4 might be needed. That was the hypothesis, as a chromatin remodeling factor at the beginning, helping to change the histone marks, but definitely also needed for the establishment of the pluripotency, as I showed you 20 minutes ago, definitely needed to maintain pluripotency. And we thought, when we now have control over OCT4 and we separate the early function from the late function, and then we will be able to destabilize the cells only when we have it at the beginning, and then stabilizing it by media cues like trapping them with transcription factors and growth factors, and hormones which are necessary to trap, for example, neural stem cells. Not pluripotent embryonic stem cell-like cells, but neural stem cells that can give rise to neurons and astroglia. And there we reasoned whether we can not undergo complete reprogramming to pluripotency, iPSCs, but to neural stem cells. And for that, we did first had a control over over OCT4 by a tetracycline responsible system, and we activated OCT4 only at the beginning of the reprogramming. And we could definitely show, yes, when we do this under those conditions, we see colonies forming that are not OCT4 positive. Yeah, so obviously we destabilize the cells and they reprogrammed into something which is not pluripotency. And we could capture these colonies, and we could analyze them in more detail, and then could show that these form neuronal markers like TOJ1. And these colonies can be also brought into adherent cell culture, and we see here in D and E passage 0, 0, and 1, and they are positive for bona fides new stem cell markers such as OLIG2, BLPP, Vimentin, PAC6, very homogeneously. They can be cultured for many, many passages in cell culture. And maybe I skipped this here. Here we did a transcriptome-wide analysis in order to check to which extent our induced neural stem cells, as we call them, INSCs, differ from the MEF at the beginning. And as you can see, there's a lot of difference in panel A between INSC line 2 and the MEF cells, whereas where we compare the same INSC2 line with neural stem cells coming from primary tissue that can be captured from E14 embryos, there's very rare genes which are discriminate between both populations of cells. So meaning that here also shown by heat map analysis that our induced neural stem cells really represent a phenotype which is more or less similar or identical to normal natural neural stem cells. And that was the first example to get also somatic stem cells directly converted by, as you see, a modified Yamanaka approach. And that results in the interpretation of this kind of scheme that everybody of you know, I guess, from basic lectures, the Waddington's epigenetic landscape, named after the evolutionary biologist from UK who analyzed the interdependence of epigenetic changes and the plasticity of cell types during development, and claiming that you start with pluripotent stem cells that can form every sort of cell type, and upon differentiation they go down these valleys and form different cell types, and usually they can go no more up. They cannot de-differentiate from the somatic tissues into the stem cell tissues. But we know from Yamanaka that it's doable. by the right combination of factors. And when we modify this in a rational manner, we can even go directly to other cell types. So from one valley to another valley, either it might be other stem cells, somatic stem cells, as in this case that I showed you, or directly converted to other postmitotic mature cells, which might be of interest from dermal fibroblasts, for example, directly to postmitotic neurons. And the concept behind that is this dual ambivalence of cell fates. On the one hand, during this conversion, the epigenetic instability needs to be induced. That results then in destabilized various intermediates, which are not really stable and need to be stabilized by either media conditions and or lineage-specific factors who define the lineage at transcription factor level, as you would see them also, for example, also under normal development conditions. That means Yamanaka type is, so getting iPSCs, we see extrinsic factors which are necessary to develop embryonic stem cells, and this is ECS medium, which is LIF medium, STAT3, YAG-STAT3 activation that in this media composition results in out of this not stable intermediates to stabilize those which form iPS cells. And when we apply this with neural medium, as we did, just by destabilizing at the beginning with OCT4, but then leaving out OCT4 at the end, we will get neural progenitor cells, NPCs. And the question arises, can we extend this concept even more? Yeah? Can we get other cell types as well? And we have then two different modalities. We have the increase of epigenetic instability that we need factors for, and we need the potential of differentiation to be increased by lineage-specific factors. And it turns out that for the epigenetic instability, one needs to use so-called pioneer factors. And pioneer factors are transcription factors which are able to bind a silent chromatin as such. So independent of the repressive marks that you usually have in completely heterochromatic, so totally silent chromatin, which is usually the case in fully differentiated mature somatic cells. It's no more accessible for transcription factors, with the only exception of pioneer factors. They can bind to it, and they a little bit able to de-repress, or in other words, make this chromatin a little bit more fluent and accessible. And then in combination with the second set of the lineage-specific factors here shown in green, you can then play the normal transcription program for the respective desired cell types. So always a combination of red and blue. And the major insight now of this mechanistic studies from Yamanaka's reprogramming was that, for example, OCT4 has two functions. Yeah? OCT4 is red and green because it's a chromatin remodeling factor. It's being involved in recruiting chromatin remodeling factors to heterochromatin, but it's definitely also part of the pluripotency transcriptional gene regulatory network. So it combines both functions, but the red function is more at the beginning of the reprogramming, and the green time was at the end of the reprogramming. But here it was the one of the examples where you have a combination of both activities and one factors. But usually it's not the case, but you can now consider that you rationally combine pioneer factors here shown in red with a particular selection of lineage-specific factors here shown in black, and the combination of both then can give you a route to directly convert fibroblasts to angioblasts, macrophages, cardiomyocytes in the mesoderm, but also endodermal cell types like hepatocytes, here primarily with FOXA and GATA4 as a pioneer factor, and in the ectodermal system, turns out that SOX2 and ACL1 has privileged pioneer functions that allows you now to turn fibroblast cells directly into, for example, glutamatergic neurons or dopaminergic neurons. So this means classical nowadays reprogramming protocols follow these lines, that you start with initial cells, you know where you want to end up with, and you need now to define the red and the green guys, which you see at the bottom. So the lineage-specific transcription factors that are being anticipated from the expression levels from, for example, mouse models, and then the corresponding pioneer factors, which need to be carefully selected and then combined together and with the corresponding methods that need to be applied in order to induce the activity of the lineage-specific factors and pioneers, either by small molecules that are able to modulate signaling pathways, microRNAs, and even epigenetic regulators, together with the transgenic manipulation. You obviously can achieve any sort of outcome of directly converted cells. There's even publications on computer algorithm where you can any sort of given cell type type in, and it will give you a rational design of a cocktail of factors that's needed to generate reprogrammed cells. And here you see just in selection, which is primarily based on direct conversions of somatic cells into different neuronal subtypes by a corresponding combination of transcription factors. Yeah, so that's the world of second and third generation reprogramming, and it even comes one step further that I would like to share now within the last 5 or 10 minutes. I hope I have this time still in this round, because I really would like to show what the next steps are in reprogramming. And this scheme is very nice to follow this up. You see in green on top the mouse corresponding published studies, and on the bottom in blue the human ones, and then with the green letters those where you have the direct conversion into mature post-mitotic cells. So we just talked about the one or the other neuronal difference direct conversion that was published 2011. You see here induced motor neurons or induced dopaminergic cells. That's, by the way, a very simple approach. You just put in the cocktail of the reprogramming factors, and you get 2 weeks later a population of directly induced dopaminergic cells, which is much more faster and directly than the RPS-based approach. Yeah? And in blue letters, you see the corresponding induced precursors or stem cell populations. And I already shared this example with the induced new stem progenitor cells with you, but in the meantime, people also published other examples of many different cell types that could have been that were reprogrammed by systematic rational modification of the reprogramming cocktail. Now, next step is to do this in situ or in vivo directly. Yeah? Why transferring or converting with a complex ex vivo procedure cell type A into cell type B with a complex experimental setup, and then at least for therapeutic applications after transplantation back into the organism, getting benefit from converted cells? Why not considering whether or not the reprogramming could be carried out directly in situ, where, for example, due to disease, mutation or inflammation or autoimmunity, the corresponding desired cell type function is being lost, like in diabetes type 1. Yeah? Let's assume you have a loss of insulin-producing beta cells due to a disease, and now you try to replace them with new converted, in situ converted, reprogrammed beta cells in the pancreas. And there's lots of studies in that direction where people try to achieve this in vivo reprogramming out of pancreatic exocrine cells into endocrine insulin-producing beta cells. And that is exactly based on these considerations that I showed to you. So here, the researchers systematically checked transcription factor combinations based on these pioneer lineage-specific factors, combination thereof, an biased selection of transcription vectors, and then found out that here you see it in E, the combination of neogenin 3, PDX1, and MAFA, called abbreviated M3. You see it marked also by the red arrow, results in an upregulation of GFP positive cells that are here used as a reporter cells, as a proxy for getting insulin producing cells. And then they put this combination of transcription factors into an adenoviral vector and then treated animals in the pancreas in situ with these adenoviruses, and you see together with the normal insulin producing beta cells, obviously there are new cells being formed that first of all here are being green due to the adenovirus, but also being red because of the production of new insulin and new beta islets, so to say, which usually would be not present, as you can see on the left-hand side, for the wild type control. And then they applied this also more in diabetic models, and there are several models that you can use for that, where you can induce diabetes in mice, so by injection with streptozotocin, you can induce this, and you see here in panel D, in control non-diabetic mice, there's no more there is homeostasis of the blood glucose very normally, but that is not the case in the diabetic mice that you can see on the top. These are mice where an adenovirus with GFP only has been administered, and then you see this M3 selected combination of in vivo reprogramming factors being under control of the adenovirus overexpression, and these partially come into glucose homeostasis like the non-diabetic mice. And this is all being also accompanied by a better vascular structure that is here shown by PCAM staining in panel B, which shows that this in vivo reprogramming somehow is feasible. Unfortunately, only shown in mice thus far and not very efficient and only in a transient manner, but showing that that conversion is from a biological perspective is doable, but needs to be then maintained and be stabilized for longer periods in order to make a real clinical application out of that. Yeah, and maybe as a last viewpoint on that, also the failure or the therapy of heart failure, for example, after infarct, is being treated on the one hand on the classical ex vivo iPS approach, where people derive iPS cardiomyocytes from iPSCs ex vivo and transplant them either as cells or as patches directly on the respective tissue in order to cure after an infarct, which is partly doable and already in clinical phase 2 and 3 trials. But also here, quite recent results showed that you can do this in situ, in vivo. Same thing. In this publication, they selected a combination of transcription factors, here with GATA4 as a pioneer factor together with MEF2C and TBX5, a combination as a lentiviral construct, and that results a combination of all results that cardiac fibroblasts in situ in the myocard are being converted partly into cardiomyocyte-like phenotypes, and it's still not able that you can dramatically improve after an infarct, but you can show that the plasticity after injury is being increased by this in vivo reprogramming. Yeah, and that is here shown in this overview, the corresponding types studies in red, as you can see in mice only, in this table here. Yeah, finally, what also was being carried out in this respect is when people tried to do this in situ conversion, or one can also say in situ reprogramming, or maybe rejuvenation, because it's at the end getting more immature, naive stem cells that might then redifferentiate then after dedifferentiation and process which occurs also in regeneration in animal kingdom, and I'm sure that you will learn about that much more in other lectures of the series. So the induction of dedifferentiation to stem cells and subsequent differentiation into corresponding tissue is a major factor in regeneration, and one can indeed artificially induce this effect in mature myocard in mice, as you can see here. Yeah, in this study, people used all of a sudden not the classical cardiomyocyte conversion protocol, but a combination of the Yamanaka factors, OSKM, and said that will be also necessary and substantial to reactivate stem cell program in situ in somatic cells. And then they combined this in a transgenic mouse where they activated this by doxycycline in different experimental setups, which are being shown here, which are being designated as a so-called pre-treatment. So you induce the expression of the Yamanaka factors before you make before you model the infarct, and then you make it just in parallel, or you do it after the set of the infarct. And in all these cases, we see at least a partial recovery of the myocard, which is here by the staining in HE, so hematoxylin eosine staining in this reddish color, whereas this scar formation is in blue, which would be not beneficial for the regeneration of the heart muscle. And in C, you see very nicely that indeed we have an increase in EDU incorporating cells after only the doxycycline treatment, so when the Yamanaka cells are being treated. So there is obviously new cells are being formed, and the hypothesis, the argumentation of the authors here, that new stem cells are being formed that are able to regenerate the heart muscle, as for example, it would take place also in embryonic mouse tissue, where this formation of stem cells out of cardiac fibroblast to cardiomyocytes is being a well-described phenomenon in previous studies. And here we can artificially induce this by overexpression of doxycycline-inducible OSKM factors. And that means OSKM is able to somehow rejuvenate in an organismic level. They did it in that study in cardiomyocyte specific, but there's also studies where people do this systemically. So they aged mice, and then in the aged mice activated OSKM, and then together with some small molecules which support the reprogramming process, could show that you really see rejuvenation of many different tissues and in systemic manners here, as it's shown for skin, stomach, and kidney in these examples. And that somehow would pave the road for getting insight into the reprogramming hierarchy and networks in situ, in vivo. And if you have really full control over it, that might be a way for a really very comprehensive targeted rejuvenation of tissues after injury or disease. But one, of course, needs to get the full insight into the mechanism, because the OSKM overexpression, too much load, would then could also result in tumor formation due to the tumor forming potential of pluripotent stem cells in adult tissues. But that will be subject maybe of future studies, and maybe if in 1 year, or maybe I guess more 5 years from now, one might have more insights into that and maybe know how one can control this then at the end also to get rejuvenation in a more controlled manner, and maybe to apply this also for human therapy. Yeah, with that, I'm at the end of my lecture, and I hope I could give you a good insight into that, what reprogramming means, in particular with a focus on new methods and on neural system, and that you're now able to understand which kind of different neural cell types can be generated and how one can use it for developmental studies and maybe also for clinical applications in future. Thank you for your attention. I'm very happy to answer your questions. So please get started.